Now, at these last slides here, I put together the summary of the entire chapter. About 3 slides here. They're going to narrow down exactly what are the challenges that DNA replication has to solve and how it solved the problems. Okay, so there are basically 5 problems on DNA replication. The first problem that the polymerases will face is that here it will find the DNA that is double-stranded and is stuck together, is on the double-stranded coil, is stuck right there. How is it going to replicate this DNA if it cannot attach to a single-stranded template? So the first problem it's going to find is how to separate the DNA. And especially when you're talking about eukaryotic DNA, that on top of being double-stranded is associated with all these histones and coiled and turned and coiled again. So this is a first problem that it will face. You have to separate those strands. And then how it does that? It does that by using helicases, by unwinding the DNA, and also it will use single-stranded stabilizing proteins to keep those strands apart. So now polymerase can go in there and make the new strand. Also remember that when you denature DNA, what do you do in order to denature DNA? What do you do to it? You heat it up. Right? You want to do PCR, for example, you have to separate your DNA strands, you heat it up to 100 degrees. When you heat it up, you're adding energy to it. Now, if these proteins are going to separate the DNA, how is it that they're going to do it? They have to unwind something that is tightly packed. It needs energy. So the unwinding of the DNA and the separation from the proteins that are keeping it stable and together has to be done with some energy. So these helicases here, they do use energy to separate the strands at first. They use ATP hydrolysis. And yeah, we have a little too much detail here on how much the ATP, you don't have to know that. Second problem, the single-stranded DNA, as I mentioned, is a single for destruction, induction of cell death. Single-strand DNA present, oh, nuclease is party, let's kill it. So you cannot have it single-stranded. You have to stabilize it with single-stranded binding proteins. The official name for the prokaryotic proteins that bind to the naked DNA is single-stranded binding proteins. For the eukaryotes, is a protein that has the same function, but they call replication protein A. Here, another problem DNA polymerases will face is that it cannot synthesize DNA without the primer. It needs the free 3' OH. It's a polymerizing enzyme. It needs free 3' OH. So they solve this problem by using the RNA primase. Another problem here is that the DNA gets supercoiled when it unwinds. If you're thinking about the prokaryotic DNA that gives you a clear picture, you unwind in one side, you wind up on the other side. So you need something to relieve the tension. You need those helicases that are going to release the tension. And then in case here of the prokaryotes, they are formally called the gyrases, and in case of the eukaryotes, they are the topoisomerases. And you have topoisomerase 1 that cuts one strand, isomerase 2 that will cut the 2 strands. Just the comparison between the prokaryote and eukaryote polymerase. With the prokaryote, you have the 2 copies of DNA polymerase 3. DNA polymerase 3 has some subunits, one that does the polymerization, one that does the correcting, and the third one that pushes the other one to do the correcting. One does the job, the other one corrects the job, but it has to be pushed by the third one. And then when you have the eukaryotic, you have more or less the same idea. You don't have too much of a pushing here, but you have one that starts, the DNA polymerase alpha, that will start with the primase activity, and then you have the DNA polymerase switching between either the alpha and the epsilon or the alpha and the delta. The delta or the epsilon are going to keep on synthesizing the new DNA. Yes, the other problem, the 5th problem here, is one that we really talked a lot about today, is that the polymerase has very low affinity to the DNA itself. So how is it going to be stuck in there? It needs somebody to get it stuck in there. So that's the function of the beta clamp. In the prokaryotes, that's called the beta clamp. In the eukaryotes, it's called pCNA. Okay. I'll never forget that because that was one of the only questions that I got wrong when I was taking a course at City College, when I was taking a master's course. And this was like the main, one big question. What enhances the processivity of DNA replication? And I could not remember. It's like, what is it? What is it? I know the DNA has to be stuck. There is some protein that goes and gets. It's like, what is it? As soon as I leave the room, pCNA. Oh, I can't believe it. I forgot that. You know, I could remember the beta clamp, but they're talking about the karyotic. Okay, I'm not really going to punish you to remember pCNA, but just remember there is a clamp that goes in there and keeps polymerase in place. When it keeps the polymerase in place, this is formally called enhancing the processivity. Okay, if the clamp is not there, polymerase is just going to fall off. Polymerase does not have the little donut to keep on stuck to the single-stranded DNA. Yes, you have a question? Yes, yes. If you are talking about the, let's say the lagging strand, remember it's synthesizing backwards. When it comes to the place that is already synthesized, and it has some 5 to 3 and 3 to 5 hexanuclease activity, it can just kick out all those nucleases, the bases that are in there, and keep on synthesizing again if it has a high affinity. Now, if it has low affinity and it gets to the point that is already synthesized, it's just going to attach. Yeah. But if it has high affinity, it's just going to keep on going and synthesize again. So there are always practical functions. And this is something that is very important to keep in mind. What is it, the clamp? Well, that's the clamp. What is it doing? Oh, it's hanging on to the DNA. Now, why? Well, because if it doesn't, you cannot polymerize it. The polymerase is going to fall off. Now, how do you get the clamp there? It doesn't go there by itself because it's a donut, remember? There is no magic trick here of the ring and the stick. It needs the magician, which is the loader, to open it up, get in the DNA, and close it again. So you need the clamp loader, you need the clamp. Okay, now polymerase goes and synthesizes. Now gets to the double-stranded DNA, the one that was just synthesized. The clamp will have low affinity to that, it will fall apart, and DNA is like, oh, I don't have to stay there anymore, I'm gone. Okay, so it will detach. It will lower, no, it will lower the affinity when you have double-stranded DNA. When you have single-stranded DNA, the clamp will be there. When you find the double-stranded DNA, it will have low affinity to that, it will dissociate. Oh, this is just that problem here with how to remove the RNA primers. Remember that you cannot have a DNA-RNA hybrid for the product of your DNA replication. It's called DNA replication, it's not DNA/RNA replication. You need to have only DNA as your end product. So you need to kick out the RNA. How do you kick out the RNA? In case of prokaryotes, you have DNA polymerase 1 that will get in there, get rid of the RNA, and put in the DNA. One step, very efficient. Getting out, put this in, get this out, put this in, replaces all the RNA with the DNA. And then we'll have for eukaryotes, is also part of that eukaryotic polymerase system, RNase H1 endonuclease, which will get in there and do the same job as polymerase 1. Kick out the RNA, put in the DNA. Now remember that at the end of this RNA that got kicked out and replaced, you have that little nick here. Remember I mentioned here that you have one side that has only one phosphate, and then the other side here that has this OH group, and you don't have a leaving group here. So the polymerase at this point cannot do anything because it doesn't have the energy to seal this nick. So what it does, it falls off. It's not going to do anything. I mean, no, good luck. So what does it do? Well, it calls somebody else to come and finish the job. Somebody that can actually use some energy, like in form of ATP, and go in there, which is a DNA ligase, go in there and close up this gap. Okay, you need some form of living group that is going to interact with this and release some energy and shift some electrons around and allow for the covalent bond to be formed. Now this is just that image of your Okazaki, of your lagging strand here. You know, leading strand, no problem, everything continuous. Lagging strand, you have to do the trick of flipping this DNA around So you can do that, extension, grabbing, going. It's just to have a clear picture of that. And then what the factors that enhance processivity, the pCNA, and in case of the prokaryotes, clamp, beta clamp. And it's just a schematic picture of what telomerase is doing at the end of the chromosome, at the end of the DNA, when you have the problem that there is no DNA to connect to. So it recognizes the sequence. This is the telomerase. It gets to the end, recognizes the end of it, and it will add those new nucleotides here, using itself as the template. This is a very neat thing about this telomerase. It uses itself as the template for the synthesis of new DNA at this end over here.